welcome to OmicsLogic 6-day hands-on online workshop on Introduction to RNA-Seq Data Analysis. This comprehensive workshop is designed for students, researchers, and working professionals in life sciences, biotechnology, and bioinformatics fields who are looking to enhance their skills in RNA-Seq data analysis. Participants will gain a thorough understanding of RNA-Seq from pre-processing raw data to advanced topics like differential gene expression, pathway, and network analysis. Let's take a closer look at what you will learn throughout this workshop. We start with the basics. What is RNA-Seq and why does it matter? Plus, you'll get hands-on experience with quality control tools like FastQC to ensure your data is reliable. Next, participants will learn how to trim and filter reads to remove low-quality bases and adapter sequences. You will then learn to align the clean reads to a reference genome using tools like STAR or high-throughput. In the third session, participants will delve into the process of read counting and gene expression quantification. You will learn to use tools like feature counts or HT-seq to count the number of reads that map to each gene and generate count matrices for further analysis. Curious about which genes are behaving differently under certain conditions? In the fourth session, participants will learn how to identify genes that are differentially expressed between different conditions using tools like DSEQ2 or EdgeR. In the fifth session, participants will explore pathway and network analysis. You will learn to use tools like 𝐃𝐚𝐭𝐞 and Reactome to identify pathways that are enriched in differentially expressed genes and construct gene interaction networks using tools like cytoscape. The final session will be dedicated to practical application. Participants will perform hands-on pathway and network analysis using RNA-Seq data. Don't miss this opportunity to enhance your skills in RNA-Seq data analysis. Whether you are a seasoned researcher or just starting, this workshop is tailored for you. Click the link in description and you will be taken to the workshop page where you can click on Buy Now to proceed to the checkout. See you in the workshop.